So we're going to talk a little bit about bacteria. Let's see if I get my pen away. So bacteria is important to biotechnology because we make a lot of products using bacteria. They're easy to manipulate. It's easy to get to the DNA. So bacteria are from a group called prokaryotes, if you remember. Right? What prokaryote means is no true nucleus. So we have our DNA in a nucleoid region, right? So it's just kind of sitting there. They have one circular chromosome, and that circular chromosome right? So it produces all of the normal function proteins. Right? So everything that bacteria is designed to do, right, will be done with that one circular chromosome. Additionally, it has plasmids, right? So these plasmids are what we're interested in. So the plasmids are what we're interested in. So these plasmids are small circular chromosomes, right? Right, with genes for survival. So it may be an enzyme that will help it digest a different type of food source. It could be antibiotic resistance, right? It could be a whole bunch of different things. The ability to form little endospores, right? Little dehydrated bacteria that will hydrate later. So it's these things, right? And they can be exchanged. When conditions are harsh. So let's say that you get a bacterial infection, then you get a high fever, right? That high fever is designed to weaken the bacterial infection, right? If a bacteria has plasmids and they have a fertilization factor called the F factor, it will seek to give its plasmids, it'll replicate its plasmids and seek to give them to other bacteria, right? It's a pretty cool survival technique because if it could keep one other one alive, then that one can make millions in 24 hours, right? So the species survives. All right, so the plasma membrane. Remember, all cells are surrounded by a plasma membrane, right? So the plasma membrane, remember, is essentially just phospholipids. Proteins. Right. So the bacteria have something that we wouldn't have around ours. So the membrane is closed in a protein peptidoglycan. So you have to remember this peptidoglycan, right here, right? It's surrounded by, and the amount of peptidoglycan we use for identification, right? So this is used. Right, so it's one of the ways we identify. So peptidoglycan, that's this purple layer here. Right, it's a purple layer here. Right. It's a protein. It makes up the cell wall of bacteria. Right? Plants have cell walls. This is the cell wall of the bacteria. We don't. We just have plasma membranes. Right. So gram-positive has a thick layer. Right. So if you look at this one, this is our positive. Right. Because Gram's iodine will stain it purple. Okay. Gram-negative, right, has a thin layer. Right. So if you look at gram-negative, a thin layer. Right. But gram-negative also has different things. Right. Gram-negative, or most often, right, we find that there's most often pathogenic. It has a lipopolysaccharides. Remember, lipo means fat, and polysaccharide means sugar. So it's a sugary fat, and it's gooey, and it's meant to stick to things. right? It also produces kind of that foul-tasting taste of bacteria when you have like a pulmonary infection, you cough up some stuff, right? Those would be those lipid polysaccharides. So these are pili right here. So pili are going to be a couple different things, but right here. So these are her pili. Right? Those are used for attaching, right? So sticking to things. Or they can make flagella, right, here for motility. Right? It's going to move it in a whip-like action. Right? Bacteria shapes, we can classify things based on shapes. You need to know what they are. So coccus means circle. Coccobacillus, it's a little bit more like, so the two main shapes are cocci and bacilli. So this is other, if you've got more than one, it's C, O, C. All right. If you have more than one bacillus, it's bacilli. All right. Could be vibrio, which is kind of like a little crescent. Spirillum or a spirochete. I'll keep spirillum and spirochete the same. Right? And then we can put modifiers on it. So diplococci, right, they're in pairs of two. Staphylo, clusters like grapes. Strepto, in a line. Right? So if I had diplobacilli, right, it would be two of these stuck together. Okay, so streptobacilli would be a whole chain of them, right? We can classify them by how they grow, right? So an aerobe, right, only grows in the presence of oxygen or air. Right? An obligate aerobe must have oxygen to survive. Facultative is that word you need to know, right? Grows better with oxygen. But still will grow without it, right? So E. coli is probably the most common bacteria we use. It's a facultative aerobe. Means it will grow with oxygen. It grows best when it's got oxygen, but it will still grow if it doesn't. Right? It invades the intestinal tracts. Right? It makes you sick when it's in your intestines because it doesn't die. It continues to grow. It just grows better out here. So you're sick with an E. coli infection. You go to the restroom and you check your hair in the mirror and you go and make someone's salad. Right? That E. coli will start growing quickly on their salad. Right? When they eat it, it'll make its way down to their gut. Right? And it'll start to grow in their intestines. And make them sick, and hopefully they'll spread it to somebody else. Right? That's how it works. An anaerobe is without oxygen. A facultative anaerobe would mean it grows best without, but will grow with. Psychrophilic, best in cold. Meso means middle. Right? So most bacterial pathogens are mesophilic. So these are most human pathogens. Right, because we're 37 degrees Celsius. And then thermophilic, right, best at temperatures 45 to 80, right, which becomes important because we'll talk about that later. Thermus aquaticus, we get our Taq polymerase from, and that's going to be really important. It makes DNA at high temperatures. You have halophiles, right, which grow best in a salt environment. So if I was going to take, so this is called a deep tube, we'll talk about, or a stab tube. Right? And I take a needle, right, and I get some bacteria out and I stick it into that growth medium, and where my bacteria grows, right, will determine what it is. If it grows only at the surface, it's an obligate aerobe. Right? If it grows only at the bottom, it's an obligate anaerobe. If it grows all over, right, It's a facultative anaerobe. You could say this is a facultative aerobe because it grows best at the surface, but it grows all throughout. Right? Aerotolerant anaerobe, it grows equally throughout. All right, so growing bacteria, so you need to know growth medium. Agars are most common. It's a solid medium. That's when we make our plates, we grow bacteria on our plates. Those are agar plates. And it's derived from marine algae. Now we can put different nutrients in it. If I'm going to grow lactobacillus, I put some lactose in it. Right? So whatever I can put in it, but that semi-solid that I grow bacteria on, right, would be like a petri dish. That's our dish that isolates colonies. Make sure you know what each does. Our slants. Right? So these are slants right here. Right? So a slant is just meant to start a colony. Right? So where I get my starter bacteria, then I'll take my slant and then I'll put it onto a petri dish for isolation. Right? Deep tubes. Let's see if I can get my pen back. Alright, so deep tube is to check for air requirements, like I told you, right? And for motility as well. Right. Okay. So based on physical state, I can have liquid medium, right? So it'd be like LP broth. Right, Lauria bertani. The solid medium would be our agar, and the semi-solid would be different. Okay, so culturing. This is my Lauria bertani. It's the most common. And really what this is, Lauria bertani just has the nutrients involved for bacterial growth. So if we were in lab right now, we would be growing broth right now. So that would be something we do. Right, so it's this color. So it's a kind of an amber color. A little bit. So if I get my pen back. So as I try to get my pen back. Hold on. Let me try to get my pen. All right. So when I just started, it'll be this. Oh my goodness, I'm having problems there. Alright, so when I just started, it'll be this color, right? This kind of clear amber color. As it grows, it'll become what's called turbid, right? It'll become cloudy. Right? Over time, it'll get sediment. Those are just dead cells on the bottom. Right? So it grows large amounts of colonies. Growth is recorded in turbidity, and we'll measure turbidity with a spectrophotometer. If you remember from chemistry, hopefully we got to this before we went out on, right? So spectrophotometer measures the amount of light that passes through a sample. So the more turbidity, the less light, the greater the growth, right? So this will just measure the amount of growth, but it won't measure the type of bacteria it is. Remember, type, we're going to do our gram stain. We're going to look at our shape morphology. We could do DNA barcoding, which we'll talk about later. Right, differential growth mediums, right, just does what they say. It could be a chromogenic medium. So chromogenic uses color, because chrome means color, right? So this is a chromogenic medium, right? So I have Staph aureus, right? It grows. I could get Staph aureus on here would be no growth, right? Or I could have E. coli and Salmonella, and this is chromogenic right here, because it's showing you that it grows a different color based on what it is, right? Or I could have my broth. This is chromogenic down here at the bottom as well. It's a chromogenic broth. Right? So, or we could use antibiotic selection, right? So we use a lot of antibiotic mediums. So when I genetically design bacteria, I'll put an antibiotic-resistant gene in the bacteria, and then I'll grow it on an agar that has antibiotic in it. So all the ones that have been successfully changed will survive, and every other one should hopefully die. Right? And that's how I can start to isolate my experiments. We could use a Kirby-Bauer test, Kirby-Bauer disc diffusion test. And what that is each disc is soaked with a different antibiotic. So this one right here would be my control. Which I would just soak with DI water, right? Or it would just be dry paper, right? And then the greater the zone, this would be the zone of inhibition. So the greater the zone, the more effective. Right, so we'll do this in the hospital as well. So in the hospital, we can do our gram stain. We can do a morphology. So if I know that this is Staph aureus, right, we have a antibiotic-resistant type called methicillin-resistant Staph aureus. You might have heard of MRSA before, right? So they'll take and they'll grow a culture, right, of the Staph aureus that we found with our patient, and they'll put discs on it, right, and they'll see what kind of bacteria kind of controls it. There's one bacteria that controls it called vancomycin, right? So that would be our vancomycin probably. All right. So we said that we use agar plates for isolation. So streak plating, right? We use it to isolate a single colony, right? And we would have done this a few times by now. But what I do essentially is I take a loop, right? So a bacterial loop, I'll put a drop of it on there, and then I'll just kind of keep going in and out. And as I do that, I just kind of thin my sample. And once I get to here, I should have thinned it enough where I'll get individual colonies. Like this is what I would want to take off and study in the future, because that represents a single bacteria that grew into a single colony, right? Right, so I could take that and I could do the same thing to make sure I have. So I'll take that bacteria, I'll grow it in broth for 24 hours, and I'll go ahead and streak it again. So serial streak plates are even better, right? But I can isolate single species of bacteria using streak plates. And the last is I can do serial dilutions, right, to reduce bacteria number, but to also count. So each colony, right, each of those bumps was called a CFU, or a colony forming unit, right? So I can take my stock, right, and I can do a serial dilution. And the beauty of a serial dilution is if I had to try to count how many colonies are on this plate, that would be impossible, right? So that colony was made from a culture. So I could take my stock culture, right, which is full strength, and I'll take, well, I'll do microliters, right, instead of milliliters, right? So I'll take 100, so if I take my example down here, I'll have 100 microliters, right? You can take 10 microliters out and have 90, or I can just use milliliters here, right? So I take a milliliter of this stock and I put it in here and I have 9 milliliters there, right? And I'll just continue to do it. I'll take 1 milliliter out of here and add it with 9 milliliters of DI water. So this is DI water, or this is broth. Right? So I take 1 milliliter of stock and add it to 9 milliliters of broth. I'll take one milliliter of number 1 and I'll put it into number 2 that has 9 milliliters of broth. I'll do the same thing. So this is 4 serial dilutions. So the calculation for this, right? So at the end of 4, so 4 would be this one right here, right? So this is my first dilution, second, third, 4. Right? Then I would try to count the number of colonies here, right? So the example I give you here is if I did a 5th dilution, right? We often do 5. This is our 5th dilution here. If I had to count the different dots, those are all CFUs, or colony forming units, right? I'd get 21. So I get 21, and each of these is a power of 10. So I did 5 dilutions times 10 to the 5, right? I transferred 100 microliters, right, from this into my plate. So I'll put 100 microliters there, and then I'll put it up to a liter, right? So that's times 1,000, right? And from there, I'll get the number of CF, or the number of bacteria. All right. So if you think about it, 1,000 is 10 to the 3rd, right? So I've got 21. Times 10 to the 8 bacteria per milliliter. Is my, that's a lot, right? So if I did this now, I could use my spectrophotometer, right? And I could do densities, right? So I could do my amount of transmittance of light through that, and then I could start to make a scale so I wouldn't have to do this every time. So I use this to coordinate my spectrophotometer. All right, well, it was a little long, but hopefully you got some good stuff. Talk to you soon.